Welcome to the PubMed Daily Review for May 5th, 2026. Today we scanned 349 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, diverse clinical studies and outcomes. Varied clinical studies investigate interventions, risk factors, and disease mechanisms impacting diverse health outcomes. This cluster covers 58 papers. Representative work includes randomized controlled trial of elective induction of labor at 40 and 41 weeks to prevent prolonged pregnancy, the effect of motivational interviewing on internet addiction and digital game addiction in adolescents, a randomized controlled trial, and trends in adverse pregnancy outcomes associated with post-traumatic stress disorder, a population-based study of delivery hospitalizations. Trend 2 of 5, pediatric disease management and treatment. The papers explore diverse clinical strategies, interventions, and diagnostic methods to enhance pediatric disease management and treatment. This cluster covers 67 papers. Representative work includes early detection of acute and early-onset cancer therapy-related cardiac dysfunction in children with cancer using a multi-parametric approach, methodological aspects of the early study, successful management of a huge pulmonary hydatid cyst in a young child, a case report, and clinical management of antibiotic drug exposure during therapeutic plasma exchange. Trend 3 of 5, clinical tools, materials, and risk. This trend develops and evaluates clinical tools and materials to assess, predict, and mitigate patient risk for improved outcomes. This cluster covers 72 papers. Representative work includes evaluating modified early warning score compliance to minimize unnecessary intensive care unit admissions, a descriptive cross-sectional needs assessment at a university teaching tertiary care center to inform quality improvement implementation, osteogenic potential of a 3-phase strontium and silicon-doped tricalcium silicate cement on dental pulp stem cells, an in vitro study, and development and validation of a clinical risk score for predicting postpartum hemorrhage in women with antepartum hemorrhage. Trend 4 of 5, cancer biology and therapeutics research. This trend explores cancer's molecular mechanisms and develops innovative therapeutic strategies for diverse malignancies. This cluster covers 103 papers. Representative work includes the silent link, exploring the impact of periodontal diseases on head and neck carcinogenesis, Tumor-Immune-On-Chip to evaluate pathophysiological feature of T lymphocytes expanded from patient tumors and lymph node tissues, and vitamin K2 extends lifespan by alleviating mitochondrial stress via the JNK 1/SIR 2.1/DF 16 signaling axis in Cenorhabditis elegans. Trend 5 of 5, health determinants and data science. These papers leverage data science to analyze diverse biological, environmental, and social determinants of health. This cluster covers 49 papers. Representative work includes loss of chromosome Y associates with altered immune cell trajectories and exon activation features, from home to hallways, how adverse childhood experiences and school context shape student relationship development, and anonymization and visualization of health data and biomarkers. Up next, our first paper out of 5, exploring evolutionary trajectories of drug resistance by Lin Feng Hu and Aryo Warshel from A Department of Chemistry, University of Southern California, Los Angeles, California, 90089. Let's break down its key contributions. Drug resistance is a persistent and significant obstacle in our fight against viral infections. To overcome this, we urgently need better ways to predict how viruses will evolve under drug pressure. This paper explores whether a sophisticated statistical model can help us anticipate these evolutionary trajectories and guide the development of more robust antiviral therapies. Researchers utilized a maximum entropy statistical model, or MAXENT, to analyze drug resistance in viral proteases. They applied this technique to both HIV and HCV proteases, which are critical targets for antiviral drugs. The goal was to see if MAXENT, by capturing evolutionary constraints, could effectively filter potential resistance pathways and help identify therapies that are more resilient to viral escape. In HIV protease, the study found that both resistance and vitality correlated with MAXENT statistical energy, but also with the number of mutated positions, which was a confounding factor. However, in HCV protease, resistance substitutions clustered within a narrow, low MAXENT range, allowing MAXENT to effectively filter evolutionary pathways. This capability combined with drug-specific vitality trends, proved instrumental in identifying therapies more resilient to viral escape. This research highlights the power of statistical models like MASLD in understanding and predicting viral drug resistance. While challenges exist, such as accounting for confounding factors, the ability to filter evolutionary pathways is a significant step forward. Ultimately, this work provides a promising framework that can inform the rational design of new antiviral drugs, helping us develop treatments that are more resilient to the ever-evolving threat of drug resistance. Up next, our second paper out of 5, Sequential ICI plus Anti-VEGF/TKI with on-demand TACE beyond RECIST progression improves pathological response, resectability, and survival in unresectable HCC by Masatoshi Kudo. Let's break down its key contributions. Unresectable hepatocellular carcinoma remains a significant challenge, with many patients experiencing disease progression even with the latest immune checkpoint inhibitor-based treatments. This paper explores a novel strategy, combining immune checkpoint inhibitors, anti-VEGF/TKI agents, and transarterial chemoembolization, or TACE, in a sequential manner. We'll delve into how this approach could revolutionize treatment by improving pathological response, increasing resectability, and ultimately extending survival for these patients. This paper proposes a strategic shift in treating unresectable HCC. Instead of stopping treatment at the first sign of RECIST progression, the strategy involves continuing immune checkpoint inhibitors and anti-VEGF/TKI agents, combined with on-demand TACE, until the disease truly becomes untreatable by TACE. This approach aims to maximize deep responses and enable curative conversion, leveraging the synergistic effects of these therapies to overcome resistance and improve long-term outcomes. Supporting this strategy, studies like the Chinese cohort by Zhao et al. Showed remarkable improvements. Patients receiving triplet therapy continued until untraceable progression, achieved an overall survival of 26 months compared to 20 months in the control group. Their progression-free survival was also significantly longer at 14 months versus 9 months, with objective response rates reaching 66.1%. Furthermore, other studies have demonstrated high curative conversion rates, with up to 35% of patients achieving a complete clinical response, highlighting the transformative potential of this sequential approach. This paper highlights a crucial paradigm shift in treating unresectable HCC, moving beyond rigid RECIST progression criteria. By continuing sequential triplet therapy until true untreatable progression, we can achieve deeper, more durable responses, significantly improve resectability, and ultimately extend patient survival. This approach calls for new clinical trial designs and endpoints that better reflect real-world practice and the full potential of these combined therapies, offering a new beacon of hope for patients with advanced liver cancer. Up next, our third paper out of 5, FibroAgent, an agentic AI tool for liver fibrosis screening and clinical decision support in metabolic dysfunction-associated steatotic liver disease, MASLD, by Basilane J. and Solomon Jayaba, from Medicine, Yale School of Medicine, New Haven, USA. Let's break down its key contributions. Metabolic dysfunction-associated steatotic liver disease, or MASLD, affects a quarter of adults worldwide, yet advanced liver fibrosis often goes undetected in primary care. Current screening methods, like static risk calculators, fall short. They provide just a number, without explaining why or offering clear next steps. This paper introduces FibroAgent, an innovative AI tool designed to bridge this critical gap and revolutionize how we screen for liver fibrosis. The team developed FibroAgent, a conversational agentic AI framework. It integrates a validated XGBoost prediction model with SHAP for patient-specific explanations, risk-stratified clinical pathways, and an interactive knowledge base. This autonomous agent uses just 7 common clinical parameters like age, HbA1c, and platelet count. Its capabilities were demonstrated across various patient risk scenarios in a simulated batch screening cohort, showcasing its potential for real-world application. FibroAgent successfully stratified patients into rule-out, indeterminate, and rule-in categories for advanced liver fibrosis, with corresponding clinical recommendations. For high-risk patients, it accurately predicted a score of 0.916, clearly identifying platelet count, AST, and age as the top risk contributors. The system also demonstrated robust error handling, maintaining analytical continuity even with simulated failures. In a batch screening of 6 patients, it classified 2 as rule-out, 2 as indeterminate, and 2 as rule-in, proving its scalability. FibroAgent represents a significant leap forward, moving beyond static calculators to an interactive, explainable, and autonomous decision support system. This tool offers a scalable, imaging-independent solution for primary care and resource-limited settings, helping to identify liver fibrosis earlier. While currently a proof of concept, it holds immense promise for improving patient outcomes by integrating prediction, explanation, and actionable recommendations directly into clinical workflows. Future work will focus on further validation and eventual clinical deployment. Up next, our 4th paper out of 5. Dual-modality imaging enables longitudinal biodistribution profiling of intracerebroventricular CAR-T therapy in orthotopic glioma by Chunjiao Li and Nanji from Neurosurgery, Beijing Tian'an Hospital, Capital Medical University, Nancy Huan, Shilu 119, Fengtai District, Beijing 100070, China. Let's break down its key contributions. Glioblastoma is a devastating brain cancer, and CAR-T cell therapy, especially when delivered directly to the brain, holds great potential. However, we currently lack robust ways to track where these cells go in the body after administration. This makes it difficult to optimize treatment strategies and understand potential risks. Researchers developed a novel dual-modality imaging strategy to track CAR-T cells. They labeled B7, H3 targeting CAR-T cells, called TX103, with special nanoparticles visible by both NGR2 fluorescence and MRI. This allowed them to longitudinally monitor the cell's journey in an orthotopic glioma mouse model, providing both whole body and anatomical insights. The results were very promising. They achieved an impressive 83.1% labeling efficiency for the CAR-T cells, and, importantly, this labeling didn't compromise cell viability or function. In vitro, NIR2 fluorescence intensity strongly correlated with cell numbers, with an R-squared of 0.973. In vivo, the NIR2 signal also showed a positive association with CD3 plus T cell density across organs, with an R-squared of 0.552, confirming the method's accuracy in tracking actual T cell distribution. This study provides a crucial new tool for CAR-T cell research. By enabling comprehensive, longitudinal tracking of CAR-T cells in vivo, this dual-modality imaging workflow can significantly improve our understanding of how these therapies distribute. This will be vital for optimizing delivery strategies, assessing safety, and ultimately, accelerating the development of more effective CAR-T cell therapies for challenging cancers like glioblastoma. Up next, our 5th paper out of 5. Tumor-Immune-On-Chip to evaluate pathophysiological feature of T lymphocytes expanded from patient tumors and lymph node tissues by Guban Cho and Sujin Hyung from Department of Health Sciences and Technology, Samsung Advanced Institute for Health Sciences and Technology, SAIHSD, Sungkyunkwan University, and Samsung Medical Center, Seoul, Republic of Korea. Let's break down its key contributions. Cancer immunotherapy holds immense promise, but its effectiveness varies greatly among patients. A key challenge is understanding how our immune cells, specifically T lymphocytes, interact with tumors. Traditional research models often fall short, so there's a real need for advanced platforms that can mimic the patient-specific tumor microenvironment more accurately. To address this, researchers developed an innovative Tumor-Immune-On-Chip platform. This microfluidic system allowed them to co-culture patient-derived T cells, specifically tumor-infiltrating lymphocytes or lymph node T cells, with colorectal cancer tumor spheroids. Using advanced imaging and cellular analysis, they could precisely observe the dynamic interactions and cytotoxic effects of these immune cells in a controlled 3D environment. The results were quite striking. While initial observations at 24 hours showed no significant differences, by 48 hours, the tumor-infiltrating lymphocytes, or TILs, demonstrated significantly more potent cytotoxic effects. We saw a remarkable 4-fold increase in CD3 plus T cells infiltrating the tumor spheroids and a 2.1-fold higher expression of cleaved caspase 3, indicating enhanced tumor cell death, all driven by the TILs. This proof-of-concept study is a significant step forward. It establishes a powerful, patient-specific platform to study complex tumor-immune interactions, offering a new lens into why some immunotherapies work better than others. While this initial work used cells from a single patient, the methodology paves the way for future clinical applications, potentially enabling more personalized and effective cancer treatments by identifying the most potent immune cells for each individual. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.